are Abby, Olivia and Erica. Our project's titled Can We Save the Common Ash? An investigation into the salicylic and jasmonic acid interplay has in the plant pathogen response of H. fraxineus and Fraxinus excelsior.
Ash dieback is a pathogenic fungus that causes leaf loss and crown dieback. This makes the wood useless for commercial production, and this is a major issue that affects over 90% of ash trees in Ireland.
After finding a study which concluded that these two hormones have an effect on poplar trees in relation to their pathogen response, we wanted to investigate if the same would work in relation to ash dieback.
For our experiment, we made a controlled medium on which to grow the fungus. We were provided with 3 different genotypes of ash from Chagas, which ranged from trees that were extremely susceptible to partly resilient to the disease. We investigated 3 different treatments of different concentrations and ratios of jasmonic and salicylic acid.
2 weeks after we inoculated our plates, we recorded the percent growth inhibition. We did this on the same day every 2 weeks for 7 weeks to obtain our results. The percent growth inhibition formula compares the colony diameter of H. fraxineus in the control dishes to the inhibited dishes. We did this to compare by how much the JA and SA at different concentrations inhibited the growth. We calculated our results using the Kruskal-Wallis H test followed by multiple Mann-Whitney U tests to see which comparison amongst the 4 variables were statistically significant.
We did this to compare treatments within genotypes. This showed a significant difference with p-values of less than 0.0001, 0.00492 and 0.00015. Following this, multiple Mann-Whitney U tests were carried out to determine that there is a significant difference between each of the treatments for genotype 1, with treatment 2 working the best, while there is no significant difference between genotypes 2 and 3.
We then carried out another set of statistics to test if the genotype had an effect on how well the treatment worked and in which genotype the treatment performed best. In doing so, we found a statistical difference between treatment 2 when comparing genotype 1 to the other genotypes, meaning that treatment 2 performed the best for genotype 1. Since there was no significant difference between the original Mann-Whitney U test when comparing genotype 2 and 3, we can say that treatment 2 worked best overall, with the p-values of 0.00016 and 0.00101. Due to completing multiple statistical analysis separately, the Bonferroni correction was added to bring down our percentage error chance.
In conclusion, we found that treatment 2, which contained a 1:2 ratio of jasmonic to salicylic at a lower concentration, worked the best on the worst effective. All treatments worked the same for the other genotypes, meaning treatment 2 would work more efficiently as it's not dependent on the genotype. As an extension to our project, we would like to test our treatment on the tree itself, as well as potentially testing sustainable methods of harvesting jasmonic acid, as it's more expensive by source.